Good day. My name is Dushara Margetson. My research project that I'm presenting is entitled Structure-Based Virtual Screening Methods to Determine the Inhibitory Potential of Compounds Isolated from Caribbean Plants against Main Protease of Sars-Cov-2. My mentor was Dr. England Buxani, and my partner for this project was Aria Duncan. So a little bit of background information. The outbreak of the novel coronavirus disease caused by the Sars-Cov-2 virus has spread rapidly around the world. It has become the current health concern to the entire world. Currently, there are no specific drugs that have been identified or are currently available for this coronavirus disease. One strategy for developing antiviral drugs is drug designing using molecular docking. Molecular docking is a key tool in computer-assisted drug design, and it involves the study of the chemical structure of a drug. So my goal for this project was to find compounds from Caribbean plants that would be good inhibitors of the main protease in SARS-CoV-2. So in this slide, this is a figure of the main protease in the molecular docking software that I use, which is the Glide software. And right here is the PBD code, which is the latest version of the main protease that can be put into the molecular docking software. SARS-CoV-2 life cycle. In 2003, the SARS virus emerged in China from bat population and moved to animals and finally to humans. 10 years later, the MERS virus also emerged from bats, transferring in the Middle East to camels and then to humans. Recently, another coronavirus has emerged in China by way of animals in a live market. So the coronavirus contains a genome composed of a long RNA strand, which is the longest of all RNA viruses. This genome acts just like a messenger RNA when it infects a cell and directs the synthesis of two long polyproteins that include the tool that the virus needs to replicate new viruses. These polyproteins include a replication complex and two proteases. The proteases play an important role in cutting the polyproteins into more functional pieces of cells that will continue to replicate over and over again. So my research question was, are the components from the 4 Caribbean plants, which I chose then was aloe vera, lemongrass, moringa, and lignum vitae, good inhibitors of the main protease in coronavirus? I hypothesize if the docking studies performed on the components of these 4 plants against the main protease have a low glide score, then that component is a good inhibitor of the main protease. So a slide shows pictures of the Caribbean plants I have used in my research. We have aloe vera to the left, lemongrass to the right, we have the moringa plant to the far left, lignum vitae plant in the middle, and the lignum vitae bark of the wood of the tree on the right. On this slide, it showed the methodology. Step 1 was to do literature review, which was basically researching articles on this topic. Step 2 was the selection of the protein, which is the main protease, 6-LZE, and the compounds, which were from the plants shown earlier. Step 3 was to download the compounds in Maestro, and Maestro is the molecular docking software used for this research. Step 4 was to filter the compounds based on Lipinski's rule of 5. And Lipinski's rule of 5 are basically 5 properties that are set for evaluating the drug likeliness of the drug. It just gives it shows like the higher probability of the drug doing well in the industry. Step 6 was to run QuickProp to predict ADME properties of the compounds. And the ADME properties are the absorption, distribution, metabolism, and excretion of the drug molecule. And it describes the drug's passage through the body. Step 7 was to prepare the protein with the protein preparation wizard in the Glide software and generate a receptor grid. Step 8 was to dock the compounds through Glide. And step 9 was the molecular dynamics simulation on TopScorer, which was not completed. In this slide, it shows the compounds from the 4 Caribbean plants that I have chosen. There was a total of 125 compounds from distributed together from each plant. So on the left were the compounds from the lemongrass. Next was the aloe vera plant compounds, the moringa compounds, and the ligum vitae compounds on the right. So on this slide, this shows the results from my docking studies. So in the previous slide, there were 125 compounds in total, and I docked all 125 compounds twice, first in SP mode and then in XP mode, which basically the modes are, SP modes are the more, XP modes are more in depth than the SP modes. So after I dock those compounds, these were the results, and as you can see right here on this line is the main protease, main protease of the Sars-Cov-2 with a glide score of negative 8.095. Everything above this protein are good compounds, and anything below are not. So the glide score relates to delta G, Gibbs free energy. If you don't recall, Gibbs free energy, in order for it to be more spontaneous, we want delta G to be more negative. And specifically, we want delta G to be more negative than the protein that we docked. So, as you see here, aluine, which is negative 8.233, would be a good inhibitor of the 6-LZE protein. But LOC, which is negative 7.830, wouldn't be a good inhibitor of the main protease because it has a lower glide score than the main protease. And so this is the result. I blowed up the main protease protein right here, and all these other compounds are good inhibitors of this main protein. Rutin was the best because it had a glide score of rutin has a glide score of negative 11.962. And in this slide, this is a picture in the molecular docking software of the rutin and protein interaction. So right in here, green is the protein, the rutin is bonding to the protein, and so this is a more zoomed in picture of the rutin and protein interaction. The bonds that are attached to the rutin and protein consist of hydrogen bonds in the yellow, some pi interactions, and some aromatic hydrogen bonding. And this is the 3D interaction through the master software. In this picture is the 2D picture of the rutin and protein interactions. So as I said before, the rutin forms the multiple hydrogen bonds, the chain, and the main ones are the CYS145 and the HIE41, which are shown in the red circles. And both of these are the catalytic dyad of the main protease, and these are the most important dyads in the Sars-Cov-2 main protease. So these two dyads are the most important binding site residues at the active site of the interaction. And this slide, it just shows the distribution of the compounds to the original plants. In the x-axis, it just shows the plants, the 4 plants that I use, and the y-axis shows the number of compounds from each of the plants. And aloe vera had 3 compounds that were good inhibitors of the main protease, and moringa had the rest 10 compounds. So for future work, I was not able to do the molecular dynamic software, which would have helped in describing some patterns and strength and the properties of protein behavior, also drug receptor interactions, the salvation of the molecules, and the conformation changes that the protein may have undergo in various conditions with the compounds. Also, I'm finishing up a research article manuscript with my other partner who worked on this project with me. And these are my references. I also give acknowledgements to the NIH mark at UVI. Are there any questions?